Okay, hello everyone. Welcome back. This is the week 12 question answer session of the NPTEL course Fundamentals in Protein Chemistry. Okay, and I am Bias Mukherjee, a research scholar from IIT Kharagpur, Department of Bioscience and Biotechnology. And this NPTEL course is coordinated by Professor Das Gupta from the Department of Chemistry, IIT Kharagpur. Okay. So we are back and this is our last class together. So in this class, we will be discussing the topics of reactive oxygen species and the way proteins are involved in this reactive oxygen species mediated cellular damage. Okay, so when our ROS is produced in our body, the proteins are also influenced, and because of that certain activities of the proteins are hampered. So we will look at that, what kind of modification is happening, which all residues are getting affected and how. Okay, so that is one of the things that we will be discussing about. And another thing that we will be discussing about is virology. So we'll talk about 2 to 3 different types of viruses. We will talk about how viruses are propagating in the host so easily. Yes, you want to say something? If you want to say something, please, you can say. Okay, you can write in the chat box or you can just unmute yourself and talk if you're interested. Okay, if you have nothing to say, we'll carry on with the class. So we'll talk about viruses today. We will talk about reactive oxygen species and we will also talk about intrinsically disordered proteins and some other small topics here and there. All right. So let's start with the class. Okay. Right. So we'll begin with reactive oxygen species. Now, the issue with reactive oxygen species are that they are formed by, they are basically free radicals. Okay. So reactive oxygen species are chemically reactive oxygen containing molecules that are naturally produced as a byproduct of cellular metabolism. So this is a very natural process and physiological levels of these reactive oxygen species in our cell is very normal. Okay. But then the problem happens when the amount of reactive oxygen species is more than the amount of antioxidants present in the cell. So antioxidants are the one that are removing these ROS, and when there is always a very delicate balance, but when that balance is hampered, then there is cellular apoptosis and a lot of different pathways are activated and cellular death is happening. Okay, so aging and neurodegenerative diseases, these are largely seen in case of huge amount of ROS production in the cells. Okay, so at high concentrations, ROS induces oxidative stress in the cells, resulting in subsequent damage to the cellular macromolecules such as nucleic acids, membrane lipids, and cellular proteins. Exactly what is happening, how all damage can be happened, we will look in the upcoming slides. Okay, so this hydroxyl radical, hydrogen peroxide, superoxide radical, and singlet oxygen, these are all different kinds of ROS species that are getting formed through oxidation. Okay, now we will look at what happens next. So these are all getting produced in the cells. Okay, now see, most cellular ROS are generated endogenously as byproduct of mitochondrial oxidative phosphorylation reaction, which comes under your respiration, formed as intermediate of oxidoreductase enzymes and metal catalases. Metal catalase oxidation. So since oxygen atom contains two unpaired electrons in separate orbits of its outer electron shell, it is susceptible to radical formation. So the sequential reduction of oxygen through the addition of electrons leads to the formation of a number of ROS. Okay, this includes superoxide, anion, hydrogen peroxide, hydroxyl radical, hypochlorous acid, etc., nitric oxide, etc. Now, in our body, there are enzymes which prevent the presence of these ROS. Okay, so they convert these active molecules into water. For example, the superoxide dismutase enzyme. Then there is catalase enzyme, which is also very important. So see, SOD or superoxide dismutase catalyzes the dismutation of the superoxide anion into oxygen or hydrogen peroxide. And catalase is another enzyme which catalyzes the decomposition of hydrogen peroxide into water and oxygen, which are very harmless species, right? So you can expect questions from this superoxide dismutase and catalase. These are very examiner friendly topics. Okay, so read about this in details. You can expect questions from here. All right. Now we will look at the next slide. Okay, so what happens when ROS is produced? Why is ROS so feared? So be it reactive oxygen species or reactive nitrogen species, these ROS actually, they operate as messengers in normal cell signal transduction, cell signaling, gene expression, homeostasis, etc. So when they are produced in high concentration, there is oxidative damage of these biomolecules that trigger. So if you understand this properly, if ROS is involved in so many cell signaling events, in gene expression, in so many different cell cycle events, cell homeostasis events, then if anything goes wrong, of course all of these different events are going to get affected. So then you understand how many different ways the cell can be affected because of overproduction of ROS. So oxidative damage of these biomolecules can trigger cellular apoptosis, aging, and different forms of pathogenesis, including cancer, neurodegenerative diseases, diabetes, and inflammation. So ROS and its associated cellular damage, this is a very interesting area of research, and one figure is given in this slide. This is taken from a review paper, and the link to that is provided in the reference. You can go through this. So this tells you that there are so many different ways in which the cell can undergo damage because of ROS overproduction. Okay. Some of these are obviously autophagy.
My voice is not audible.
My voice is not audible. Okay, 1 second. Can you hear me now? Can you hear me now? Okay, if you still cannot hear me, I will leave the call and join once again, okay?
Now it is audible.
It is audible now? Okay. That is great. Okay, so let's carry on. Did you hear whatever I said until now? Otherwise, I can repeat. Okay, so I'll give you, I'll just repeat in a sentence or two just for your understanding. So basically, reactive oxygen species, if they are produced in very large quantity compared to the antioxidants, then what happens? Then there is a problem. A lot of different processes are hampered, and your autophagy, apoptosis, inflammatory diseases, then neurodegenerative diseases, cancer, everything is activated, everything is triggered, and the cell undergoes death. Okay, this is the main gist of whatever we talked until now. There are different enzymes like superoxide dismutase and catalase which act on these reactive oxygen species and produce harmless compounds from them, okay, like oxygen and water. Right, now we will go to the next slide. Okay, so what happens when these ROS are very high in concentration? ROS affects lipids, proteins, and DNA. Okay, To the lipids, there is chain breakage, and then there is alteration in permeability and fluidity. So that means your cell membrane is going to get affected because lipids make up the membranes, right? Organellar membranes and cellular membrane. Coming to proteins, specific amino acids can get modified. Fragmentation of the proteins can happen. Aggregation is very, very common. Aggregation can happen. Altered enzymatic function, enzyme inactivation, and increase of the enzyme susceptibility to proteolysis. So we will look at some of this in details when we study about cysteine and tyrosine mediated protein damage because of ROS activation. Alright, so all of this different things are happening, and proteins are very, very important in our cell, right? Every function in the cell is more or less carried out by proteins. So if the protein function is getting hampered, you can understand what is going to happen to the cell. Okay, now we go to DNA. What is happening in DNA? There can be breakage of the strands. There can be modifications of the bases. Okay, there is modifications, removal of the nucleotides, and then there can be cross-linking. So basically DNA damage is happening too. Now what happens? Because of all of this, you saw that so many different diseases can happen. Okay, now we will look into details what exactly happens in case of cysteine and tyrosine. Now cysteine and tyrosine are very important when it comes to acting as biomarkers. So they are often seen as biomarkers during this activated ROS production. Okay, what happens to cysteine is, cysteine initially you can see in this diagram that this SH group is present in the cysteine, right? But then because of oxidation, it can get converted into sulfonic acid, which is SOH, which is reversible oxidation. So you can recover it somehow. It can get converted into sulfonic and sulfonic acid, that is SO2H and SO3H. These are irreversible. Because of these modifications, enzyme inactivation can happen, disruption of the enzyme structure. You can understand that if cysteine is there and there is disulfide linkage, of course because of oxidation this is not possible anymore, correct? Then what is going to happen? Its 3D structure is going to like break, and what is going to happen? The activity of the enzyme is going to get affected. So structural aspect is gone, activity is also gone. So if that enzyme is involved in some kind of, let's say, regulation, the entire pathway is gone then, right? So you understand from here that sulfonic acids are a result of irreversible oxidation, and your sulfanic acid is a result of reversible oxidation of cysteine residues, and disulfide bond is no more formed. Okay, then we come to tyrosine. So tyrosine is also an important biomarker. What happens is it can get modified. Okay, it can get modified into dityrosine. The structure is given here. This is your dityrosine. Then you have nitrotyrosine. So dityrosine is formed by ROS. Nitrotyrosine is formed by reactive nitrogen species. Okay, and this they actually have different properties of absorption of light. Okay, one of them is fluorescent, the other one is not that fluorescent. So we will talk about them soon. So see, all of these different modifications are happening in tyrosine as well, and you get dinitrotyrosine, you get 3-nitrotyrosine, and you get tyrosine radical. All of these modifications are happening. All right. Now we will move on. There is a small question. You can try answering this one. Okay, you can write in the chat box. This is your last class. You already know what to do. Okay. You can write down in the chat box whatever is the answer according to you. Please write down in the chat box. So which one of the following oxidized form of cysteine is most likely to be directly linked to protein damage? All the names are very similar. So if you are able to remember, C. C is sulfonic acid form. Okay, so both of you are saying C. Let us see which is the right answer. Okay, give me a second. I'll just shut this off. Alright, so yes, C is the right answer. Okay, we saw in this particular slide that Irreversible oxidation was happening in case of sulfinic and sulfonic acid, and this is the one that we are talking about here. So this is sulfonic acid, right? And it is directly linked to protein damage. Now we come to another question. This is dityrosine and nitrotyrosine. We just saw these two species can be formed from tyrosine, right? So they are actually biomarkers. Now one of them is detected using UV visible spectroscopy. Another one is detected using fluorescence spectroscopy. Now why is that so? We will look at the next slide. So here you can try answering this. Which one do you think would be detectable better using UV visible, and which one would be detectable better using fluorescence spectroscopy, and why do you think so? A. Alright, we'll see the right answer. No, the right answer is actually C. So see what happens here is in fluorescence spectroscopy, the intensity of dityrosine. So dityrosine is formed over here, right? When two tyrosine molecules are cross-linked by a covalent bond. This is what is happening in case of dityrosine. So in case of dityrosine, actually what happens is this, one second. Yeah. So because in dityrosine, we see that the intensity is enhanced. In case of the fluorescence intensity is actually enhanced. So fluorescence spectroscopy is very useful in case of dityrosine. But then when you come to UV visible spectroscopy in case of nitrotyrosine, which is the second example that we saw in this slide, here what happens, normal tyrosine gives one small peak here, okay. But then in case of nitrotyrosine at the same pH of 3.5, you will see that there is one peak here and another peak here, correct? So this is one peak and this is the other peak. So basically you are getting two peaks for nitrotyrosine at 278 and 350 nanometer each. Okay, and therefore it is easier to detect this using UV visible spectroscopy. Now we will read about this in details, why exactly that happens. So nitrotyrosine forms when tyrosine undergoes nitration at the phenol ring, typically through interaction with the reactive nitrogen species. Okay, at 278 nanometer, it shows absorbance corresponding to the aromatic rings electron transfer, which is also seen in case of normal tyrosine. At 350, however, the nitrotyrosine exhibits unique absorbance peak caused by the nitro group, that is NO2 group that is attached to the aromatic ring. Because of this, you are getting two different peaks. Now we come to your tyrosine and dityrosine. So what happens here is tyrosine, normally you know that this is useful for UV visible spectroscopy because aromatic ring, it is having this electron localization, because of which it is absorbing and it is emitting light, right? It is absorbing the UV visible range spectrum. So now what happens is its fluorescence intensity. So tyrosine itself has some intrinsic fluorescence, and that is why it is used for fluorescence spectroscopy. Okay. Now, so fluorescence intensity enhancement in case of dityrosine, it is basically increasing. So the presence of cross-link structure in dityrosine changes its electronic environment, leading to greater absorption and greater absorption of the excitation wavelength of light and stronger emission, because of which in fluorescence spectroscopy you are able to detect and distinguish dityrosine compared to normal tyrosine. Okay, there are two rings in dityrosine, so of course you will get more emission, correct, and of course more absorption, right? So now we will go to the methods of protein sequencing. So in our last class, we were talking about this, which method you cannot use for protein sequencing, and there we came across Sanger sequencing. So one of you was saying that even in the PPT, Sanger sequencing is given, ma'am. So then why can we not use it? So The difference is that it was given as Sanger's method, and that is Sanger's sequencing. So when we say Sanger's sequencing, it is usually DNA, and here the enzyme is used, and I talked about it last day, right? But Sanger's method, the reagent is actually, so basically this method is called Sanger's method, where you are going to use this fluoro-dinitrobenzene reagent, and you are going to get this N-terminal, yeah, you got it now, right? Okay, okay. So that is what. So if it is given in the question, you have to read carefully whether it is Sanger's sequencing or Sanger's method. All right. So N-terminal protein sequencing. So for N-terminal protein sequencing, we have three methods. One of them is Sanger's method, one of them is Edmond degradation, and the third one is dancyl chloride mediated method. Okay, so all the three are given here. So we will look at this. So here everything is happening. This is your acid hydrolysis. So free amino acids are there, and this is the this like thing is actually coming bound to the amino acid, and then through different steps you will come across it. So see, in Sanger's method, this particular fluorodinitrobenzene is used. The polypeptides react with FDNP to form the dinitrophenol complex with the polypeptide, and subsequent analysis releases a colored dinitrophenol labeled amino acid, terminal amino acid. Right, so amino terminal labeled amino acid, which is then identified by characteristic migration on by using different chromatographic methods. So that is how you get to know about the N-terminal residue, okay, using Sanger's method. Then you have Edmond degradation. So all of this are taken from Leninger's and other PPTs. So I've provided the links. Go through these in details because you might get asked about these in different questions. Okay, so coming to your Edmond degradation, here phenyl isothiocyanate is used, and the final product that is formed, that is phenyl hydantoin derivative, and this is how you are going to get the N-terminal amino acid, right? So first the polypeptide is treated with phenyl isothiocyanate to form a phenyl peptidyl derivative. Then gentle hydrolysis releases the terminal amino acid as phenyl hydantoin. Then it is separated and detected spectrophotometrically. Up to this stage, the method is used to determine the N-terminal amino acid, and this method can be extended to determine the polypeptide sequence. For this, after release of the PTH, the remaining intact polypeptide is shortened by one amino acid. It is ready for another cycle. So this is how you can actually know about the entire sequence of the sequence of the entire protein, right? Lastly, we have dancyl chloride. So dancyl chloride is used to determine the N-terminal amino acid, and this is a very sensitive method, and it uses fluorescence measurements. All right, so you can read about this one too. Okay, now you try answering this question. This is a simple question, but this type of questions come in your DBT exam, and you will feel that it is a simple question because right now we studied about all of these three methods, but that time if you are not thinking correctly, there is a very high possibility that you will go wrong. Yes, D. But why is that so? For what is cyanogen bromide used? Why? Where do you use CNBR? Where do you use CNBR? We'll come across CNBR later in today's class, I think. Yes, correct. Right. Okay. Right. So we'll go to the next slide. Okay. So cyanogen bromide cuts the peptide bonds on the carboxyl end, and they are asking about N-terminal peptide sequence. Correct. So CNBR cuts at the methionine residues at the C-terminal end. So this is why it is very important that you practice the questions, because only when you practice you will be able to think instantly that okay, this is what I am supposed to answer. Right. So now we will talk about viruses for a little while. All right. Virus. Okay. I am sure everybody knows a lot about viruses after the entire COVID period. Right. So you will be able to answer this better than me. So tell me, virology is a complete subject in itself. You have to devote a lot of time to virology. It is simple, it is scoring, but then you will have to understand it very nicely, and you have to remember what virus carries what genetic material, because that is something that they always ask. Okay, what kind of virus carries what kind of genetic material? So it's something that you have to remember. Baki, if you the rest of it is very simple and logical. Okay, so basically a virus is a non-cellular infectious entity made up of genetic material that we can see here. This is the genetic material of the virus. This is a bacteriophage on your left, and on your right that is a virus. Okay, human immunodeficiency virus or HIV. Now, okay, so virus is, it lies between living and dead. I'm sure you know this, right? So inside the host it is replicating and it is showing forms of life. Outside the host it can be crystallized, it is next to non-living. So it is made up of genetic material which is either DNA or RNA. It is not both at the same time. This is a very important point when it comes to your questions. It is always either DNA or RNA. Okay. And it is having a protein coat that is covering this genetic material. Together, the protein coat and the genetic material is called the nucleocapsid. Viruses are divided into different types based on their structural configuration. Please read about that. They have icosahedral heads. Some viruses, for example, you will study about this T4 phage and all that. So they are having this icosahedral head, which means that it is made up of 20 facets and all that. So it's very important that you read about this because they ask questions, okay, about the classification of viruses. And based on this Baltimore system of classification, there are different classes of viruses. So read about them. Okay. Now surrounding this capsid protein, there is often this envelope protein. Now the viruses that are having this extra covering, they are called envelope viruses. Other viruses that are not having this envelope, they are known as naked viruses. Okay. Right. Now what gets into the host? Let's say for the bacteriophage, does the entire bacteriophage come into the host cell, or does it send only part of it into the host cell? Tell me.
Ma'am, it's only DNA.
Right,
Material.
exactly. So only the genetic material is injected into the host cell. But for this particular virus, let's say if it is an enveloped virus, it is having an envelope. This envelope is actually coming and fusing with the cell membrane. So envelope is made from the cell membrane, right? Envelope comes and fuses with the cell membrane. Rest of the virus gets in. Okay. So we will talk about lytic and lysogenic phages also. And lytic and lysogenic phages is a very, very interesting and very important and favorite topic of examiners. When you are learning about lytic and lysogenic phages, read about this Cray protein, read about your entire this what is it called? Tell me this. I'm forgetting. This genomic material basically. Okay. The entire genomic material of the lambda phage. It is very important. What is happening? What particular there are so many different promoters. So which is getting activated when? What gets activated because of, in case of lytic cycle? What gets activated in case of lysogenic cycle? The order of activation, that is very important. And they ask questions from there every time. So please read about that. Okay, so now we will be talking about lytic and lysogenic. So what is it about lytic and lysogenic? You can answer in the chat box. Who is a lysogen and who is a lytic phage? What is lysis? This you know, definitely you know. Lysis? No, lysis is not a enzyme. Lysis means to break open, right? You lyse the cell, exactly, break down, right? So lytic phages are the phages that are going to break the cell open. Yes, breakdown of the cell wall, that is cellular lysis, correct. So lytic phages are going to come into the host cell, they are going to amplify, they are going to replicate, then they are going to break open the phage and go out. Lysogenic phages are very clever. What they are going to do is they are going to come into the host cell. They are going to integrate with the genetic material of the lysogenic lysogen. So remember, lysogen is not a virus. Lysogen is the host cell. Lysogenic phage or prophage is the one that is associated with the genetic material of the host cell. That is a prophage. Okay, remember the terminology, it's very important. So it is going to stay attached to the genetic material of the host cell when the conditions are bad. So it is going to replicate with the genomic material of the host cell. When the conditions are very bad, so there is stressful condition, let's say there is less oxygen, there is more toxins in the media, so it is not getting enough food, that is called induction of it is called induction of the lytic cycle. Then what happens? It excises, it removes itself from the genetic material of the host, and then it starts synthesizing its own protein coat, its all the components of the body of the virus. Then it gets packaged, the genetic material gets packaged into the cellular coat and the entire viral particle gets formed, and then it comes out. Okay, so it can either break open the cell and come out, or it can just come out very silently without breaking open the cell. And that is the thing about viral infections. So after the virus gets into your body, there is a very latent latent phase during which the virus will not show any signs of its activity, and that is why there are so many diseases. For example, AIDS. So people, HIV virus enters into the body, it stays latent for let's say 20 years, and after that it shows signs, and then that is the time people realize that something is definitely wrong with my body. So that is why viral diseases are very difficult to detect and manage. Okay, incubation period or window period, correct, exactly, that is what it is called, incubation or window period. Now the thing is, we will talk about viral attachment and penetration. So first, this is your cell, virus comes and sits onto the cell and it attaches, right? This is called attachment. Penetration means there is kind of an injection and the genetic material is put into the host cell. Then what happens? The genetic material, there can be integration in case of lysogenic phages. So it will go and integrate into the genetic material of the host. Then what happens? It will replicate with the host. And after that what happens? There can be excision. When will it excise? When there is very stressful conditions going on. If the virus knows that the cell is possibly going to die, then it will excise. And it will use the machinery of the host to, it will take over the machinery of the host and it will start making copies of its protein coat and the other components. Then it will get packaged into the protein coat, right? And there is also some cases of recombination. So recombination means there is genetic exchange between the viral DNA and the protein DNA, host cell DNA. Because of that, you will see that in bacteria there are these attachment sites, ATT sites. So that is where recombination has happened. So viruses have been living on earth long before we even came into existence, right? So even our genetic material is having some like components of this viral genetic material. People talk about jumping genes, transposons, right? So transposons and all of these different, there are various different kinds of genetic elements in our body, in bacteria, that are said to have obtained from viruses through this course of different recombination and different processes. Okay, so that is one thing. Recombination is very common, and because of recombination, you have possibly read about transducing phages, right? This is how they amplify, multiply. Okay, then you see that there is this stressful condition, and the prophage excises, and then it replicates, and then it gets assembled and packaged into its body, and then it can lyse the cell and it can leave. So this is the entire life cycle of a lysogenic phage. If it is not a lysogen, then what will it do? If it is not a lysogen, it will come into the body of the, it will come into the host cell, and it will exist as a round prophage DNA. Otherwise, it will be integrated into the nuclear material of the DNA, right? That is lysogenic phage. And if it exists like a separate entity, it is a prophage. Okay. Now we will go into the next slide. Now, the thing about viruses is, for this, I would recommend you can read Bruce Albert's. Okay, there it's given very nicely, or you can even read the books that we always refer to. There also it's given properly. Viruses are very, very clever. So, even without having their own cellular components, how are they surviving for ages, right? They are very clever, and they have adapted because they are multiplying so quickly. They have the capability of undergoing different mutations, and then there is selection. So selection of the fittest, right? And they come across different kinds of mutations, and they are very, very prone to adaptations also. So genetic materials in viruses are of different types, and based on that, they are again divided into different classes. Okay, so usually animal viruses do not always express their genes using the normal flow of genetic information from DNA to RNA to protein, which is a central dogma of biology. Some viruses have a double-stranded DNA genome like cellular organisms and can follow the normal flow. However, some have single-stranded DNA. Some have double-stranded RNA. Some have single-stranded RNA. Okay. Now what happens? So the nature of the genome determines how the genome is replicated and expressed as the viral proteins. So if a genome is single-stranded DNA, then what is going to happen? Then host enzymes will be used to synthesize a complementary strand. Okay, so from single-stranded DNA, you will get the complementary strand, and thus producing double-stranded DNA. This double-stranded DNA is then normally propagated. Fine. If the viral genome is RNA, then what happens? There can be three types of RNA genomes. Viruses have fixed reservoirs. Reservoirs as in you can type, we'll carry on. Okay, and once you have answered, we can read it. Alright. So there are polio only in human. Yeah, we'll read about poliovirus too. I think it's there in this PPT. Okay. Oh, acha. So you're saying that they are very specific to the host. Yes, viruses are sometimes specific to the host. They are of different categories. Okay, so some viruses affect animals, they affect primates, they affect humans. Some viruses can affect these intermediary host also can be there. Okay, so there are viruses that can propagate inside, let's say, cattle, and they can also affect humans. So yes, they have their own reservoirs. Okay, so Right. So now the RNA viruses, they can have three type of genetic material. One is positive sense single-stranded RNA. Now this is a very important place. You have to understand positive and negative sense because questions will come from this part too, if you want to answer them, of course. But this viruses are very scoring. So if you can remember and if you can logically understand things, it will be easy for you to attempt the questions and score good marks. Okay. Chalo. So there can be double-stranded RNA, there can be positive single-stranded RNA, there can be negative sense single-stranded RNA. Now what is a positive and negative sense? The question that will come is, which one can form mRNA directly? Is it positive sense RNA or is it negative sense RNA? Which one can form mRNA directly? That is how the question is going to come. Okay. Now see, if a virus has single-stranded positive sense single-stranded genome, it can be translated directly to make viral proteins. So positive sense can directly form mRNA. However, if it is single-stranded negative sense, then what happens? Viral genome with positive sense single-stranded RNA acts like cellular mRNA. However, if a virus contains negative sense single-stranded RNA genome, the host ribosomes cannot translate it until the negative sense is replicated into positive sense by RNA-dependent RNA polymerase. Acha, now this RNA-dependent RNA polymerase in our body, where is it present? This you can answer in the chat box. Where is RNA-dependent RNA polymerase present in our body? Reverse transcriptase. No, reverse transcriptase is again different. That is RNA dependent DNA polymerase. This is RNA dependent RNA polymerase. Right. So RNA dependent RNA polymerase is not present in our body. Okay, this is something that the virus is going to carry. Okay, all the genes that the virus needs, if they are not present in the host, it is going to be carried by the virus. In our body, there is DNA dependent RNA polymerase, right? In our body, from DNA, mRNA is formed. So we have that enzyme. But the reverse one, that is for reverse transcriptase, we don't have it in our body. This also we do not have in our body, right? RNA dependent RNA polymerase. So the RNA dependent RNA polymerase is brought in by the virus and it can be used to make single-stranded RNA from the original single-stranded negative sense single-stranded RNA genome. So from the negative sense single-stranded RNA, you have to make complementary positive sense single-stranded RNA, and this positive sense can be then used for production of viral proteins. Okay, right. Now see, the RNA dependent RNA polymerase is also an important enzyme for the replication of double-stranded RNA viruses because it uses the negative sense strand of the double-stranded genome to create the positive sense strand. The newly synthesized single-stranded RNA copies are then translated. So this positive sense RNA is acting as your mRNA for synthesis of viral proteins. Okay, got it? Now see, in the figure they are showing a particular flow chart, right? So RNA viruses contain positive sense single-stranded RNA that can be directly read by the ribosomes to synthesize viral proteins. Viruses containing negative sense RNA must first use the negative sense RNA as a template to synthesize the positive sense RNA before synthesis of viral proteins. Okay, now we'll go to the next one. What about reverse transcriptase? You talked about reverse transcriptase just now, right? So we will look at the example of a particular virus which all of you are very familiar with. This is HIV. the very dreaded virus, human immunodeficiency virus, that is known to cause AIDS. Okay, so retroviruses, they are positive sense RNA virus like HIV, carry a special enzyme called the reverse transcriptase within the capsid that synthesizes a complementary single-stranded DNA. So positive sense RNA is there, but we need a cDNA. So cDNA is your what? Complementary DNA, right? So from this positive sense single-stranded RNA, it is creating this cDNA, and this RNA-dependent DNA polymerase, this is again brought in by the virus. We don't have it in our body. In our body, RNA never gets converted into DNA, correct? So it is brought in by the virus. Now what happens is the single-stranded DNA is then made into double-stranded DNA. So DNA-dependent DNA polymerase is obviously there in our body. So single-stranded DNA is converted into double-stranded DNA, correct? Now what happens? This DNA integrates into the host. That is how it stays latent for years and years and years, and nobody comes to know that this patient is infected. It does not show any signs of infection. Okay, so which can integrate into the host chromosome and become a permanent part of the host. The integrated viral genome is called the provirus. So you understand the difference between provirus and prophage. Prophage is the one that is present, right? So virus now can remain in the host for a long time to establish a chronic infection. The provirus stage is similar to the prophage state in the bacterial infection during the lysogenic cycle. We talked about that in the previous slide. So that was a bacteriophage, so it was being called a prophage. This is a virus, so it is being called a provirus, correct? However, unlike prophage, the provirus does not undergo excision after splicing into the genome. So see the different stages of its life cycle is given here. So HIV fuses with the host cell membrane. It is having the envelope where the glycoproteins are present, GP120 and GP I think 40. So please read about the functions of these glycoproteins. These all questions might come for your exams. So please read the functions. And then what happens? HIV RNA reverse transcriptase, integrase and other viral proteins enter the host cell. Integrase is the enzyme that is needed for the formation of the prophage stage. So genetic material of the host and the genetic material of the virus fuse together using this integrase enzyme. Then you have the viral DNA is formed by reverse transcription. Okay, viral DNA is transported across the nucleus and integrates into the host cell DNA. So reverse transcriptase, where is it acting? It is acting in the cytoplasm. Please remember that. Then it is getting transferred, viral DNA is getting transferred into the nucleus where it is binding with the host DNA as a, and this provirus stage is attained. Then the new viral RNA is used as the genomic RNA to make viral proteins. New viral RNA and proteins move to the cell surface and new immature HIV gets formed. The virus matures when the protease releases the proteins that form the mature HIV. That is how the virus buds out from the cell. Okay. Now we will look at a question. So which of these processes does not happen in HIV? Replication, reverse transcription, translation and transcription. Which of these processes do not happen in case of HIV? B, C. Okay, let's see. Yeah, the right answer is C. So So replication and transcription, both of these are not happening. So in HIV, reverse synthesis of DNA from RNA is performed by the enzyme reverse transcriptase. So DNA is getting formed, and translation is the synthesis of the protein from RNA, which is of course happening. And since mRNA carries instruction for making new viral proteins, each selection of the mRNA relates to making different parts of the HIV. Okay, so if you look at this once again, from here you can understand that reverse transcription is definitely happening, and then integration is happening, and therefore we don't see DNA replication happening as such. And then we see that translation is happening and your transcription is happening, and we are getting the viral proteins. Okay, all right. Now we come to Ebola virus. Okay, so we come to Ebola virus. Okay, so now Ebola virus is a very dangerous virus because of the fact that there are no vaccines or therapeutics against Ebola virus yet. Okay, and Ebola virus is very dangerous because the mortality rate goes up to 90%. It goes from 30 to 90%. So that means if any person is infected with Ebola, 9 out of 10 people might die. Okay, and that is why Ebola outbreaks are very scary. Another thing with Ebola is that it is a filamentous shaped virus. The dimensions are given, which are not important. It is encapsulated, so it is having a capsule. Single stranded negative RNA is present, so negative RNA. Okay, and there are 7 proteins that are expressed. The first one is your glycoprotein. This is located on the viral envelope. It's an envelope virus. Glycoprotein spikes help the virus to attach to the host. There is VP40, which is viral protein 40, membrane associated protein that block the immune responses and play a role in the viral assembly and budding. Okay, nucleocapsid, so structure composed of the nucleoproteins NP that encapsulate the viral genome. VP30, a phosphoprotein involved in RNA transcription. So now if you're thinking whether you have to remember all this VP40, 24 and 30, yes you have to. And their functions too, yes you have to. That is why for virology you have to remember certain things. It is the way it is, okay. Viral genome. So single-stranded negative sense RNA which codes the genetic information of the virus. And of course, you have VP35, which is a nucleocapsid protein that inhibits the antiviral immune responses. Polymerase protein, an enzyme that catalyzes RNA replication. Of course, this is a negative sense RNA. So you will have to prepare this single-stranded RNA, right? Positive sense. And the enzyme is RNA-dependent RNA polymerase. So you have this polymerase protein for RNA replication. Now, we go to the next slide. Okay, so see NP is essential for RNA encapsulation, GP is glycoprotein, important for attachment and entry into the host cell, VP24 essential for viral assembling and transcription by being a part of the nucleocapsid structure, VP30 suppression of the viral RNA silencing, and VP35 binds to NP, NP is your nucleoprotein, to remove the nucleocapsid and facilitate the transcriptional expression. VP40 required for viral localization out of the host cell membrane and gives the filamentous shape to the virus together with the glycoprotein and helps maintain the structural integrity. Here you talk about Ebola, the deadliness of Ebola. Okay, so Ebola is incurable and deadly. The outbreak in West Africa in 2014 had happened where nearly 24,000 people were infected with Ebola, and out of them more than 10,000 people died. So no approved treatments or vaccines for Ebola are available. Okay. Now here comes in the BSL labs. So biosafety level. Now when you're working with organisms that are pathogenic or infectious, there are different safety levels. Okay. For example, I think we work with biosafety level 2. Biosafety level 1 and 2, these are non-pathogenic. Biosafety level 3, these are organisms that are dangerous, that are pathogenic, and they can maybe spread through blood. And for example, if anyone is working with hepatitis B, they are working with, let's say, coronavirus. Okay. But then the ones that are biosafety level 4, so there are no vaccines available against them, and the mortality rate is very high. So those come under biosafety level 4. And there are different kinds of biosafety cabinets, and you can read about this in India also. There are different places in which biosafety level 3 labs are there. For example, IISc has biosafety level 3 at like this thing, and they work with mycobacterium species. Okay. And there is biosafety level 3 cabinets and lab facilities. There are also biosafety level 4, but that is very rare in India. You can read about that, it is very interesting. And questions are also asked from here too. Okay, biosafety and the different kinds of cabinets, and how the air pressure is regulated, how entry of air and exit of the air from the room is regulated. Then the people have to wear gloves and everything, and they have to wear face mask and everything when you're working in these biosafety cabinets. So that is some important topic that you can go through if you're interested. Right. So now we come to this question. This is possibly the, oh, we have a few more questions after this. Okay, try to answer this one. The nucleocapsid of, okay, so which is true about Ebola. Nucleocapsid of Ebola virus contains a DNA genome. The lethal virus is made up of 7 proteins encoded by its genome. Matrix proteins help to adapt different shapes of the viruses for driving the budding process. Nucleocapsid of the virus is circular in nature. Which is true? So you're saying that 2 is true. Okay, let's see. Okay, so 2 and 3 are correct, and C tells us that nucleocapsid is not circular, it is cylindrical. Correct. And it has 7 proteins. That is true. So this statement 2 is correct. Matrix proteins give the virus shape and drives the process of budding. So this is also correct. And Ebola has negative sense single stranded RNA, so it does not have a DNA genome. Therefore 1 and 4 are wrong. Okay. Now we come to these proteases. An unknown protease cleaves the peptide sequence given here into 3 polypeptides. The polypeptides are also given. Okay. Polypeptides are also given. Which protease has been used? C. C is chymotrypsin. Okay, let's see. C. So chymotrypsin selectively cleaves the carboxyl amino acid tryptophan, tyrosine, and phenylalanine. And so one of the issues here is that I am not sure if chymotrypsin cleaves when this proline is here on the right hand side. It is not supposed to cleave when proline is there on the right hand side. Okay, so you can actually look into this. Here it will definitely cut, but I don't think it can cut here. Okay. So hence in this peptide it cleaves the amide bond between phenylalanine and proline, and also cleaves between tyrosine and glycine. Okay, we go to the next one. Now we have 2 more examples. You can try these two. So the first polypeptide has been cleaved by thrombin and trypsin. What are the digested fragments that you will get? Where will thrombin cleave and where will trypsin cleave? So it's very important that you remember the cleavage sites of all of these enzymes. F followed by proline is not cleaved. Exactly. So F followed by proline cannot be cleaved, and therefore in this question it would only be two fragments. This one full fragment will come and this one fragment will come. Correct? So I think this is not correct. Oh, sorry. Yeah, you try answering these two questions. In the next question, we have tryptic digest of an octapeptide where you're getting 2 lysine, 2 methionine. For the first one, you're saying that it is C. Okay. The tryptic digest means that trypsin has been used to digest a particular octapeptide, and you're getting 2 lysines, 2 methionine and 1 serine and 1 proline and 1 glycine and 1 arginine yielded 2 tripeptides and 1 dipeptide. Both the tripeptides do not contain arginine. Okay. But there is only one arginine and it is there in the dipeptide. So which of the sequences is correct? So this is very interesting and these questions will take a little bit of time, but then all these questions are very nice to solve. Try solving this one. C. Okay, let's see. Let's try to. So for the first one, it is C. So since thrombin cleaves carboxyl side of arginine and trypsin cleaves the carboxyl side of both lysine and arginine residues not followed by proline, digested fragments are this and this. Okay. Now for this one, you see that trypsin cleaves the carboxyl side of both lysine and arginine residues not followed by proline. Correct. So after tryptic digestion, the peptide fragments are this, this and this. So both tripeptides do not contain any arginine. And here are the places where the cut had happened. Okay. This is how you can get back the sequence of the octapeptide. Okay. Right. So this is already solved. We can go through this quickly. Okay. This is already solved. So which one of the following amino acids is preferably present in the intrinsically disordered protein and play an important role? Oh, acha, acha. So for this, you need to look into this first. So we'll talk about intrinsically disordered proteins. This is our last topic for the day. Now, the thing is, not all proteins are folded. Until now, we have been talking about stable proteins, folded proteins. How do we make proteins stable, right? But then some proteins are made unstable and disordered by nature for their definite purpose, right? So people say God's plan. So even these proteins are disordered and they are not very well folded, and that is also basically done by nature because of a definite purpose. Okay. So these intrinsically disordered proteins, they are highly specific to the sequence, DNA sequence they bind. If we are talking about, let's say, DNA binding proteins, many of them are disordered and they are highly specific to the DNA sequence they bind. Because of their flexibility, they can undergo conformational change very quickly, and this phenomenon of autoinhibition is also very prominent in case of these disordered proteins. Okay. Now we will look into these disordered proteins. Okay. So intrinsically disordered proteins, there is one very interesting thing about intrinsically disordered proteins. So one thing is specificity, the other thing is affinity. Okay. They bind to they can bind to different DNA sequences of very similar sequence, but they only bind to only one specific region in DNA with very high sequence specificity. Okay, how is that happening? So that is because there is always this kind of equilibrium going on. There is always a very delicate balance maintained between specificity and affinity. Okay. Now, this disordered proteins, they are having very long C-terminal and N-terminal arms, which are very flexible. This is actually providing them the energy. This is making the conformational changes energetically favorable. Okay, so here is a method in which these disordered proteins, partially unfolded proteins, they are binding to their target partner, and this mechanism is called fly casting mechanism. So fly casting comes from the analogy of a fisherman casting a fly fishing line into water. So a loosely extended line can cover more area than a coil or a stiff line, making it easier to catch fish. So even when the target is far off, this N-terminal and C-terminal long extended regions go and bind to the target DNA quickly and easily, okay, by spending less amount of energy. So similarly, IDPs or proteins that are unfolded, partially disordered, they can explore a larger area of conformational space while searching for their target protein. Okay, so conformational space means that they can explore different conformational states. So different structural conformations can be attained, and since they are already disordered, it is favored. Okay. So mechanism is increased capture radius. So it means that the capture radius is larger. The disordered state of an IDP allows it to extend and sample a larger area, increasing the likelihood of encountering its target. This is analogous to the fishing line. Okay. Rapid binding. Once the IDP comes into close proximity with its target, it can rapidly collapse and fold. So this is called folding upon binding. So once it comes very close to the target, it folds into a specific structure, induced fit, and it matches the binding site. Correct? Folding upon binding or induced fit. This leads to an efficient interaction. An example of the fly casting mechanism can be seen in case of interaction between transcription factors and DNA. Many transcription factors have intrinsically disordered regions that help them to locate specific DNA sequences. The extended flexible structure of transcription factors allow it to scan large regions of DNA very quickly. Once the correct binding site is attained, this disordered region becomes more ordered, folds into a stable conformation, forms a very strong interaction with DNA. Got it? Now we will look into this question. Yes, this one. So the question is, which of the following amino acids are preferably present in the intrinsically disordered proteins and play a role in preventing the formation of stable structure? Which of these proteins do you think would be very important in the intrinsically disordered proteins? Definitely the proteins, the amino acids that are very flexible. So, your what? Glutamic acid, proline and arginine, they are very largely seen in the intrinsically disordered protein. So, proline is a helix bender, correct? And when proline is present in a peptide sequence, it will disrupt the secondary structure. In the presence of polar amino acids, stable non-polar core structure gets prevented. Hence, the option A matches all of these different criteria. Okay. Now we can go to the next one. Which of the following is not true? These are two questions, and these are the last two questions of our course. So please try to answer these two questions and then we'll end the session. Which among the following is not true about intrinsically disordered proteins? So IDPs are proteins which lack stable three-dimensional structure. IDPs are disordered only in localized regions. IDPs carry low densities of charged amino acid residues. IDPs exist in highly flexible conformations. Which of these are not true? Okay. So B, you're saying this is for question number 1 or question number 2? One is B, okay. And what about question number 2, which is incorrect for the fly casting mechanism of IDP? Specific binding site is searched for and found while folding, reaches a high affinity conformation. Non-specific binding site searched for and found while folding. So, okay. Easy sampling of many conformations. So, I have got my answers. Let's see. Yes. So, in both the cases, C is the right answer. Right? That is because IDPs carry low density of charged amino acids. That is not true. IDPs carry high density of polar and charged amino acids such as lysine, glutamine, and serine, arginine, and glutamic acid. Fly casting mechanism of IDP, specific binding site is searched for, found, and folded. The protein gets folded. Okay. Thank you so much. These are your different resources that you can avail. So whatever questions are there, whatever answers are there, whatever topics I have discussed today, all the links are provided here. So please go through them. Thank you so much for all these months. Thank you for attending my classes. Thank you for cooperating. Thank you for asking so many questions. With you, I have also learned so many things. Thank you so much to all of you. I hope I have been able to. Yeah.
Ma'am, app study materials गुड़ाक आसुनी.
Can you say
Hello.
again? हाँ बोलो.
मैम apps में जो study materials थे वो आ नहीं रहा है अब.
Study materials.
वो आ नहीं रहा है जो PDFs थे lectures के वो अभी available नहीं है.
अवेलेबल नहीं
apps
है.
में.
कौन से? मेरी ये
जी
वाले
मैम.
पीपीटी आपको नहीं मिल रहा?
नहीं दूसरे वाले जो हमारे मैम के जो है.
अच्छा, मैम के पीपीटीज नहीं मिल रहे. आप एक काम कीजिए, यू कैन मेल द एनपीटीएल टीम. ठीक है, वो लोग देख लेंगे ये चीज. Previous year's questions you can see, you can get YouTube videos, you can search online, you will get.
No, ma'am, there is no any previous question on YouTube or NPTEL website. Please organize any mock paper which will possible for NPTEL site before the examination.
Before examination.
And examination. Any mock paper.
Right.
I will just practice.
I will talk to them. Okay, if they're allowing, then we can do that. Otherwise, I will just upload a mock your test paper kind of thing. Then you can practice from there. Okay.
Yes, ma'am.
And all the questions that we have practiced until now, these are also mostly your previous year's questions only. Okay. So all of these are also previous year's questions. So if you go through them properly, if you practice the topics that we covered in all of these questions, that should also be helpful to you. But yeah, I mean, definitely we can do that. Okay, I'll talk to the team. Anything else that you want to tell me? Otherwise, we'll end the session. We'll end the class. Thank you so much.
Thank you so much, ma'am.
Thank you. Okay. So let's end the session then. All the best for your exams. All the best in life. Okay. I'm ending the call for everyone.
Thank you, ma'am.